The broadcast is now starting. All attendees are in listen-only mode.
Hi, thank you for joining me today as I discuss the ISO-18562 series updates, a CRO point of view. My name is Nate Frederick, and I am a toxicologist with Nelson Laboratories. I currently write toxicological risk assessments from the chemistry testing of medical devices, and I help leverage the biocompatibility safety for end users of these medical devices. Once again, thank you for joining me today as we'll be taking a deeper look into this 18562 series of updates that were published earlier this year. Alongside these updates, we will be looking at what these new standards bring to the table in terms of if they provide the toxicologist, laboratory, or medical device manufacturers any additional tools. If you have seen my previous webinars in the 18562 series, this first part may be a bit of a review. We're just going to get started with the basics of the standards. As we're all familiar with these standards, I'll quickly talk about the brief history and then move on to the updates. Over the previous decades, the ISO 10993 International Standard Series has been the overarching set of standards that helps provide guidance on how to evaluate medical devices for biocompatibility. However, there's been a little bit of a split in these overarching documents because of special externally communicating devices, such as those that carry air or gas to the lungs. When a gas is the carrier of the externally communicating device, the risk profile is a little different than if the carrier was a liquid, which is the suggested matrices of the ISO 10993 series. From a toxicological standpoint, more often than not, these gases can be more toxic, more problematic, and in some cases, they can result in unsafe exposure to chemicals that come off of medical devices. To see how these standards are a bit different, the 10993 suite are the standards that cover all testing under biological evaluation of medical devices, whereas the ISO 18562 series looks at the gas pathways within healthcare applications. The use of these two series of standards goes hand in hand with various FDA guidances to further support the safety of all medical devices. As an introduction to ISO 18562, the main goal of this series is intended to be a guidance to determine the potential biological risks, such as looking at the risks of the materials and processes to the patient. For the risk management stance, ISO 18562-1 is responsible for the overarching risk management process of these potentially indirect, externally communicating devices. While the categorization is something that most of us are familiar with, it won't hurt just to briefly touch on it so we're all on the same page. The term indirect contact is used for a device or device components in which a fluid or gas pass through before coming into physical contact with the body or body tissues. We can then look at this as the device component. It does not physically contact the body tissue. Furthermore, according to ISO 10993-1, this direct tissue contact will lead to insufficient assessments and testing of gas pathway devices. This is essentially where ISO 18562 takes over. As an overlook, the four standards that we will be looking more into are 18562-1, the evaluation and testing within a risk management process, 18562-2, testing for the emission of particulate matter, 18562-3, testing for the emission of volatile organic substances, which will often be referred to as VOSs, and lastly, 18562-4, testing for leachables and condensate. So right away, we are already introduced to the first big change, the naming of volatile organic compounds to volatile organic substances. Before we get into changes in its entirety, it would be nice to go over where the FDA stands in terms of acceptance. Luckily for us, the FDA accepts in full 18562-2, 18562-3, and 18562-4, whereas the FDA has only partially recognized 18562-1 because of two clauses. Within 18562-1, it is stated that parenteral toxicological information may be used in the absence of inhalation toxicological information without modification. However, we know as toxicologists this is not the case. From the toxicologist side, when using parenteral toxicological information in lieu of inhalation, we just have to add an uncertainty factor to account for the differences in routes of exposure. By the FDA not accepting this clause, nothing essentially changes when the inhalation toxicological information does not exist. In the regulatory pathway, from the first initial conception steps, the biological evaluation, to regulatory submission, these standards would more than likely follow in this order. A strong BEP should be authored. It follows guidance from 18562-1 and 10993-1. This plan lays out the outline of what testing may be needed. From this outline, we'll proceed with the applicable chemistry testing, and this can include testing for volatile organic substances, leachables, and condensate if the device has the opportunity to produce humidified gas or condensate which may reach the patient, and then of course, testing for particulate matter. After the applicable chemistry testing has been conducted, a toxicological risk assessment may be authored, which ultimately provides margin of safety calculations for each chemical constituent seen. From the TRA, a biological evaluation report may be of use, and this combines all the testing into one document to ease submission review. Once all these steps are followed, the regulatory submission is ready. However, there is one update that may not look as straightforward as it seems. Within ISO 18562-1, there is this flowchart. Now, we're only looking at the latter half of this flowchart, and right in the middle, it mentions that you must prepare a biological evaluation report. However, there's really no mention of a toxicological risk assessment, which may confuse some sponsors. If they strictly aim for this BEP before having a tox risk assessment authored, they may have to backtrack and get that tox risk assessment completed. Now that we know the background of the standards and how they play a role with overall regulatory submission, let's go ahead and get into the main changes. Within 18562-1, there's maybe 15 new definitions. However, this was a lengthy list. I've just picked out a few of the most important that I think everyone who works with the standard should have a general idea of. A good one to start out with would be abnormal use. I've included the definition as taken straight from the standard on the slide, but abnormal use is really how the device should not be used, like eating cereal out of a CPAP mask, just something that is beyond the reasonable means of what the device is used for. Now, use error is a little bit different. There's more leniency here. Maybe the IFU states that the device can be used for 10 hours a day, no more. However, a patient uses it and it may continue to use it for 12 hours a day. This could be a use error that falls within normal use, and these use errors should be taken into account in the applicable risk assessments. I have included this figure 1 from 18562-1 to help further show that use error falls under normal use and may be taken into account or even brought into question with regulators. Exposure dose is essentially the amount of chemical constituent that can reach the body. Exposure dose is mainly in condensate. If a patient can swallow, I suppose exposure dose is more of an oral exposure. Inhalation dose is essentially the same as this exposure dose, but it's the amount of VOSs that may be inhaled. Well, tolerable exposure is on this list, but it's nothing that's new. I left it on here as it is important to note that this term did not change. Some of us are familiar with the updates in ISO 10993-17. It excludes the use of TEs. However, within 18562 series, this term is still sticking around. Lastly on my list, we have VOSs, volatile organic substances, and this is further broken down into volatile organic compounds, very volatile organic compounds, and semi-volatile organic compounds. The boiling point of all these compounds can help separate them into different categories. While not necessarily terms, there's a slight change to the classifications. Limited and prolonged exposure is staying the same. However, for medical devices coming into contact greater than 30 days, permanent contact has been changed to long-term exposure. This has since been updated to match the 10993 series. Within the changes of ISO 18562-1, patient categories and body masses were more defined. 2 additional patient categories were added, as well as the age range for each categories. These categories help better define which patients may use whichever medical devices is at hand. This can be on the basis of body weight or age, and it helps the toxicologist accurately derive tolerable exposure or tolerable intakes for the various categories. To go along with the body mass changes, new breathing volumes were introduced. Rather than the 20 meter cube per day for adults, it is now assumed to be almost half that, at 11.5 meters cubed per day. However, as we are introduced exercise ventilation volume, this is a new concept. In this case, if a patient is assumed to be on the exercise ventilation for up to 3 hours a day, we would assume the daily breathing volume of 11.5 meters cubed for 21 hours, and then exercise ventilation for remaining 3 hours. This requires a more in-depth calculation to derive the overall meters cubed per day. And as we can see now, there is, a much more defined breathing volume scheme, and it does make us as toxicologists our job a little easier. We can now more accurately calculate the inhalation dose. Dealing with the threshold of toxicological concern, this may seem a little more easier and straightforward. For devices that have limited or prolonged contact duration, the 120 microgram TTC from ICH M7 will be used. For devices with long-term exposure, the applicable TTC will be derived based upon the breathing volume. In this case, for a device that may be used across all patient categories, there would be up to 6 different TTCs that are derived. To determine the TTC for each patient category, we just multiply the breathing volume by 2, which is defined here in the TTC in microgram per day column on the right-hand side. This derivation is still protected on most chemicals. However, special care needs to be taken to screen out any cohort of concern chemicals. There's really no notable changes to ISO 18562-2, as particulates still must be tested for all air pathway devices. Most of the options stay the same. There's three test options: a single particle filter or a double particle filter, inertial particle separator, or a particle counter. Particulates are tested by air being passed through and around the device and then being collected and ultimately counted. This air must be of a certain flow rate and temperature. Particulate testing requires the highest clinically relevant flow rate, or the highest flow rate for which the medical device may be set at. The highest flow rate must be used when sampling, as it is seen as a more worst-case scenario. Of this air that has been sampled, particulate matter 2.5 and particulate matter 10 is then analyzed and taken into account within the toxicological risk assessment. This particulate testing does not account for any nanoparticles coming off the device. Per the US EPA 40 CFR Part 50, it has now been adopted into the ISO 18562-2, and these are the limits for the respective particulate matter size, excluding nanoparticles. 12 microgram per meter cubed for 2.5 micron particulate matter, and 150 microgram per meter cubed for 10 micron particulate matter. From a toxicology standpoint, this size does matter, as the small particles are more toxic than the larger ones for multiple reasons. The 2.5 particulates can be suspended into the air for longer periods. This increases the chances of them being breathed in. Once they're breathed in, they have the opportunity to penetrate further into the lungs alveoli. This is where gas exchange happens. From here, they could also enter the bloodstream, and health effects from these small particulates can range from the development of asthma and bronchitis to heart attacks and strokes. Moving on to the volatile organic substances, ISO 18562-3, this is the test required for all gas pathway devices. Here, the devices are tested in a similar fashion as particulates. Clean, dry air or nitrogen may be pushed through. If the air is tested just through the device, it only tests the interior of the device. Devices can also be sampled with air going through and around, which tests both the interior and exterior of the device. The flow rate for this testing differs from particulate testing. We need to look at the lowest clinically relevant flow rate. If, for example, the lowest flow rate of your device is 0.5 liters per minute, it may not be a bad idea to test at 0.1 liters per minute. The gold standard in gas pathway medical device testing is the TO15 method, or referred to as the EPA canister method, which Nelson Labs has adopted. In this method, air samples are collected in a canister made out of stainless steel, as pictured. However, there's more than one method that are also allowed per the new standard, such as the sorbent tube method. Once the samples are collected, the air is then analyzed using GCMS, which then identifies and quantifies the analyte seen. We are then left with a list of quantitative and semi-quantitative compounds, targeted and non-targeted compounds. The length of time for this VOC sampling should be multiple 24-hour periods. It is typical for sampling to occur in 3 separate 24-hour periods, even for long-term devices. This gives us 3 whole days worth of data. The first 24 to 48 hours, 48 to 72 hours, and then we could, look at any sort of release kinetics from the chemicals seen. However, if there are expected to be a lot of volatiles to come off the device, it may be a benefit to increase sampling out to 3 or 4 days. As for the notable changes for ISO 18562-3, this includes some of my favorite changes in this battery of standards. Within and accepted by the FDA, it states that if VOSs are detected at compounds below 1 microgram per meter cubed for targeted or 2 microgram per meter cubed for non-targeted compounds, they do not need to be reported or even assessed within the toxicological risk assessment. This cuts down on the amount of time the analytical chemist has to spend identifying peaks and cuts down the toxicologist's amount of work needed to find relevant toxicological information. There's no inherent risk by making these levels a cutoff. This amount is very small and it is below the threshold of toxicological concern after the changes. Keep in mind that the TTC was 2 microgram per meter cubed times the breathing volume. The next major change to the standard is it is now suggested that aldehyde testing should take place. It is stated that specific assessments for formaldehyde or other carbonyl compounds shall be included. While there really is no extensive list of the aldehydes that are required, here's a few examples of which aldehydes may be captured within this assessment. Formaldehyde and acetaldehyde are the main ones we believe. This standard also states that if the targeted analysis for this aldehyde testing takes place, we may also omit these compounds below the 1 microgram per meter cubed as discussed previously. This aldehyde testing, it really should require no additional sampling, as the lab doing the analysis can use the other sampled air to perform this analysis if they're qualified. Another change that has been made is the addition of certain verbiage which helps determine the amount of time testing may need to be performed for the device. In this standard, and as seen in previous standards, they give us this nice decay curve, figure 1 in ISO 18562-3. This is the decay curve of emission of volatile organic substances. It is expected that most VOSs are released as soon as the device is new and taken out of the packaging. I'm sure we have all experienced this as we get something new, open it up, and are hit by all the volatiles coming off. It's almost like that new car smell. It doesn't last. As these medical devices are sampled, we are expecting that exact same thing. More VOSs right away, and then going down over time, or going down over the sampling periods. As it is hard to explicitly know how many VOSs are going to come off of the device when designing a study, it may be beneficial to do multiple days of testing, 3 or 4. However, within this new standard, it states that when the first 24-hour test returns a very low value, below that allowed for longer-term use, then further tests need not to be performed. Essentially, if all the compounds seen on the second day of testing, marked by a 3 on this decay curve, are below the calculated tolerable exposure values within the tox risk assessment, no further testing is needed, and that last sampling period is not needed. However, as mentioned earlier, this is very hard to pinpoint at the start of testing, and if for some reason on that second sampling period is above that TE value, another device may need to be sampled for that 3-day period. Now, moving on to ISO 18562-4. While it's not a notable change to the standard, it's now accepted by the FDA, meaning we can actually follow this standard of submitting gas pathway devices that may generate condensate. Within this standard, it outlines how to determine if leachable testing in condensate is absolutely necessary, and if it is, the approach that must be taken. This is through chemical characterization. Once that is completed, calculation of the exposure dose, as well as the correlating risk assessment in terms of the TRA. For a device to be considered for leachables, it must be considered if condensate may be formed. Not only does condensate have to form, but it has to reach the patient. This must be experimentally determined, and it can be done in the lab or even with sponsors, but it has to be adequately documented. Normal use conditions, however, a worst case parameter must be used. Temperature must be taken into account, and then ultimately the amount of condensate formed and has the ability to reach the patient in 24 hours must be recorded. If this condensate formation is less than 0.1 mils in 24 hours, no further testing is necessary. Whereas anything greater than 0.1 mil would warrant that leachable testing. So now, given a device that will generate condensate, E&L testing will be needed. To begin with, the devices must be extracted to generate that chemical profile. These extractions follow conditions as outlined in 10993-18. The devices can be extracted by full immersion, as seen on the far right, or the device could just be gas pathway filled and capped, as seen on the left. Each of these extractions are acceptable. However, when the device is submerged, we will get an assumed exaggerated amount of extractables detected due to both the gas pathway and the external surfaces of the device being extracted. Because of this, it is a more worst-case extractables profile. However, these results may also be able to be scaled accordingly. For example, if the internal and external surface areas are exactly the same, and they make up 50-50 of the device, the results seen may be scaled by half. There are various approaches that may be used for the extraction. However, most devices are normally extracted for 72 hours at 50 degrees Celsius. These gas pathway devices are extracted in water, which is only mentioned in 18562-4. However, if there are drugs or anesthetics used, ethanol, IPA, or other relevant solvents may be used as an additional extraction solvent, but it's not in the scope of 18562-4. Once we have extracted the device, we can then analyze our extraction fluid using a variety of analytical techniques. Essentially here, we're looking for a large variety of compounds within the extract. The substances we could be identifying are either inorganic or organic based. Our ICP-MS or ICP-OES will pick up any metals or elements. These are the non-carbon based compounds. To detect organics or carbon-based compounds, our GCMS will pick up any volatile organics or semi-volatile organics. These will be slightly different from the volatiles seen in the gas pathway testing. Lastly, we can identify any non-volatile organic compounds with the LCMS. All of the analytical sensitivity of this testing must be documented appropriately, as well as the relative response factors, internal standards used, and the correct analytical evaluation threshold derived from the applicable dose-based threshold. Now, there's quite a bit of finite detail that goes into this E&L studies, so please don't hesitate to reach out if help is needed to design one. Before I get into some case studies and examples, let's not forget about inorganic gases. While it's not covered in the 18562 series now, the standard does state this series does not currently address the biocompatibility hazards associated with the following substances being added to the respirable gas stream. Nonetheless, when applicable, some authorities having jurisdiction require the manufacturer to evaluate the following, with the following being ozone, NO2, CO2, and CO. So, as a medical device manufacturer, or a CRO, we must always be on the lookout for various devices that are intended to perform a chemical reaction, that have UV for disinfection, or any devices with motors or moving parts. This testing may be easy to overlook, and we just have to catch it ahead of time. Once we have generated results, we can then see what compounds or analytes come off the device, and we can then assess everything all at once with the toxicological risk assessment. We want a relevant toxicological analysis, and I must emphasize on relevant as our end goal is patient safety. Within toxicological risk assessment, we want to include a device description, categorization, explanation of testing, and then the assessment of all the analytes seen. We assess the particulates, the VOSs, the inorganics, and condensate through E&L in these TRAs. A lot of behind-the-scenes work goes on here. This is where the chemistry report may undergo grouping, so that like compounds are summed together for a total exposure to the patient. From this, we also derive various tolerable exposure or tolerable intake values and provide a margin of safety. We then have a short calculation discussion so it can be seen how each value was derived. Additionally, if there are various compounds that are known to have adverse toxicological effects, further information or some sort of risk mitigation may be needed. While a tox risk assessment is quite challenging, the basic steps really are not. You generate your chemistry results for your medical devices, and this is done with the previously discussed robust testing. Additionally, from the results, these compounds may be grouped or assessed separately. And then research available toxicological data for each compound from literature reviews. From these reviews, we try to look for no adverse effects level or lowest adverse effect level. This is typically referred to as the point of departure. This point of departure is then computed into a TI and TE using various uncertainty factors and then further calculated into a margin of safety. These point of departures are researched through literature reviews of trusted databases and sources such as PubMed, ECA, ToxNet, ATS-DR, and IRIS. While not spending too much time on the toxicological risk assessment side of things, the margin of safety is essentially the end goal for each compound. This is a way to see if said compound is safe. To calculate the margin of safety accurately, we start off by deriving a tolerable intake value. To begin, a no adverse effect level or any other relevant point of departure can be derived from literature. It is then divided by the uncertainty factors. These uncertainty factors can be different each time a TI is derived. The typical uncertainty factors are based on the inter-individual variations between humans, the extrapolation between humans and the animal that was used in the toxicological study, and the quality and relevance of the experimental data. Additional uncertainty factors can be added as the toxicologist sees fit. After taking these uncertainty factors into account, we are left with the concentration microgram per meter cubed. The TI is then further calculated into a tolerable exposure. Since we are dealing with the gas pathway device, we will use the inhalation rate multiplied by the tolerable intake to derive that tolerable exposure in microgram per day. Once we have this tolerable exposure, the margin of safety is a fairly easy calculation. We are just looking at the ratio of the tolerable exposure to the chemical constituent seen. Any margin of safety less than 1 is considered a toxicological hazard. Now, we can look into the specifics about how the standard has changed. I have included a real sample data set here. This methodology used was a quantitative analysis. These are all targeted compounds. In the second column, microgram per meter cubed, this was the highest amount detected across multiple sampling periods. From this highest amount detected, the patient exposure in microgram per day is calculated using the new breathing volumes as listed on the left. This gives us the inhalation exposure for every single compound. If authoring a toxicological risk assessment, it may not be necessary to derive the exposure in terms of microgram per day for every patient population, just the population that may be able to use the device. If you have a keen eye, you can see that some of the compounds that have a concentration less than what 18562-1 states are included, and that is 2 microgram per meter cubed for this quantitative compound. I kept these in this data set as you can see how the new standard plays a role. We can see that hexane, freon, difluorylmethane, and O-xylene are all below the 2 microgram threshold for this quantitative method and can now be omitted from the chemistry report as well as the toxicological risk assessment. As a conservative approach, heptane and methylene chloride, the compounds detected right at 2 microgram per meter cubed, were left in. While this update saves us time from assessing 4 compounds, we can spend more time on the compounds that may need additional toxicological research. Here is another sample data set that we have seen. Previously, we would have to assess all 5 of these compounds. Now, hexane, heptane, methylene chloride, and difluorylmethane are omitted, leaving us with just acetone to assess. I think this data set is a great example of the time save that this new standard allows us. When these standards were first released, internally we had some initial concerns, such as omitting everything below 1 microgram per meter cubed and 2 microgram per meter cubed. Is this toxically relevant and safe? Well, the new TTC aspect within the 18562 series multiplies 2 by the breathing volume. So anything that will be omitted will ultimately be below the TTC. Of course, an experienced toxicologist still should go through the results in full and see if any omitted compounds are members of the cohort of concern or if there are any highly potent or toxicants that stick out. Additionally, when we keep in mind that we are extrapolating from a low sampling flow rate to higher patient breathing volume to calculate this worst case exposure, this should also give us some sort of protectiveness. Additionally, most toxicological risk assessments assume that there are new devices used daily and that they are used all day long, which just further helps patient protectiveness. While 18562-4 helps lay out condensate testing, what if the gas pathway can be used in anesthesia or some sort of drug or drug component? The standard only lays out water and then references 10993-18. This could be confusing for some in the industry as now the list of standards to test and assess with the toxicological risk assessment are ever growing. Choosing the most appropriate solvent tends to be a challenge. When drug products are introduced to the device, the polarity of the extraction needs to be mimicked to the best available solvent, all while showing that worst case extraction. As you can see here, I have listed the various polarity indexes of common laboratory solvents. This is taken from ISO 10993-18. For medical devices that will come into contact with drug products, we do suggest that 10993-18 is followed. However, it's not straightforward and may need help of a chemist or a toxicologist to help drive that study. Now that SVOCs are included in volatile organic substances, how can we be sure that these are being omitted from the device? SVOCs have a lower volatility when compared to VOCs, and they evaporate more slowly and have that higher boiling point. Using the analytical techniques at hand, is it further unlikely that they're being volatilized when sampled? Or can these SVOCs be a source of contamination in the manufacturing process? If that's the case, wouldn't these be picked up or taken care of through a cleaning validation? Lastly, well, not a concern, but more of a thought process. With aldehyde testing being added to the array of tests, is this relevant for all devices, or is this adding to the burden of medical device manufacturers? Are we able to omit aldehyde testing in general if a thorough paper assessment on materials and construction happen? If certain adhesives used as solvents or binding are used, then aldehyde testing, yeah, it should be completed. But if a device has a safe material history where aldehyde release is basically impossible, can this testing be omitted? Before I leave you all today, there's a few additional considerations that must be kept in mind. Further analytical testing may be needed to help demonstrate safety of the device. If not enough data points are created to show safety through the toxicological risk assessment, more sampling periods may be needed. If there's a material change or a manufacturing process change in your device from what has been tested previously, this additional testing may also be needed on that new device. If components touch the skin of a patient, they could be dual classified as an indirect breathing pathway as well as a surface device for skin contact. A great example of a device like this would be a CPAP mask that fits on the face of a patient. This must undergo the big three tests: cytotoxicity, irritation, and skin sensitization, as these endpoints are not covered within the risk assessment. Lastly, with these changes, do any devices currently on the market need to go through additional testing? The answer is simple. No, devices that are currently on the market do not have more testing, and as stated in ISO 18562-1, this series of documents shall not be used to mandate retesting of historical products assessed previously using appropriate addition to this document at the time of the assessment. I want to thank each and every one of you for attending this webinar. Please keep in mind that Nelson Laboratories has much more than just your toxicological needs. We have experts that specialize in packaging, sterilization, and biocompatibility, in addition to the hundreds of laboratory tests. Also, I have included my email, and please do not hesitate to reach out with any questions or further comments. Thank you for attending this webinar today.